So elongation of translation is going to continue adding one amino acid at a time, right? So you're going to have your tRNA bringing in the next amino acid, hop on in there into the A site, codon matches with the anticodon. You have peptide bond formation to elongate the polypeptide chain by one amino acid. The whole chain gets handed off then to that first transfer RNA. The ribosome then shifts one space over. This empty tRNA goes into the exit site and leaves, which you see here. And then the tRNA that's holding on to that growing polypeptide moves into the P site and allows then the A site to be open for the next tRNA to come in. Now, as we get into termination of translation, once the bottom of this A site contains a stop codon. Remember, stop codons don't code for amino acids. They're simply just coding for a function, for the stop function, to tell the ribosome that, hey, you know, at this point, you're done. So once a stop codon appears there at the bottom of the A site, there's no tRNA that has the correct anticodon to match that codon. So what happens instead is one of these release factors, a protein, comes in there, binds to the A site where the stop codon is, and the ribosome really kind of doesn't know any better, and it still tries to sort of add the next amino acid in line, but there is no amino acid, right? Because we have the release factor in there instead of a transfer RNA with an amino acid. And so the ribosome tries to add this non-existent amino acid and instead ends up cutting off the polypeptide from the existing tRNA in the P site, and it releases the polypeptide. So the polypeptide is now free, can go on to fold and become a functional protein, and the tRNA is released there from the P site. Once that happens, along with a little bit of energy required here, the rest of the ribosome falls apart, releases the release factor, releases the mRNA, and then the mRNA either might get degraded or it might get picked up by another small subunit of the same or a different ribosome, and the whole process can continue all over again. All right, so let's talk a little bit about the polypeptide itself. So once it gets released, or even maybe before it gets released, it actually already starts to fold a little bit. So that sort of chain of amino acids that's hanging off the outside of the ribosome can start to take on its final shape. Remember that we talked about how the gene of a protein determines its primary structure, so the sequence of amino acids, and then the sequence of amino acids, or its primary structure, in turn determines its shape. For some proteins, just making the polypeptide and having it fold does not make a fully functional protein. Sometimes they need to be modified. So there are some post-translational modifications that can occur to polypeptide before it becomes a functional protein. This can include cutting of the polypeptide. So insulin, for example, is produced as proinsulin, and then it needs to be cut before it can become the actual insulin hormone. Other proteins might need carbohydrate attachments or lipid attachments added to the protein itself before it can become functional. And again, a lot of that takes place, for example, in the endomembrane system, in the ER or in the Golgi apparatus. Now let's briefly talk about the ribosomes, right? We mentioned earlier in the semester that there are really two kinds of ribosomes, and they're different based on their location, right? You've got your free ribosomes that are just floating around in the cytosol, and the free ribosomes make a certain type of protein. They make the proteins that are meant to stay in the cytosol and function in the cytosol of the cell. Then you've got the bound ribosomes. These are going to be either bound to the rough ER or the nuclear envelope, and they make a different kind of protein. They're going to make those proteins that either, number one, are meant to participate in cellular membranes, so plasma membrane or organelle membranes, or number two, those proteins that are meant to be secreted outside of the cell. Now, ribosomes are identical, and they can switch between being free and bound. So let's take a look at how this might happen. This slide looks really complicated, but it just gives you sort of a stepwise view of how, for example, a ribosome initiates translation, and if it's meant to make a protein that, let's say, needs to be secreted from the cell via the endomembrane system, how it then becomes a bound ribosome. So we'll start on the far left-hand side. All right, so number one here. we've got in the cytosol a ribosome. The small subunit has found itself an mRNA here. It grabbed a hold of that mRNA and it started translation once it found itself also the large subunit. So you can see here the polypeptide is hanging out here out of the end of the ribosome. Now, this particular mRNA that the ribosome grabbed here, this is an mRNA that is meant to be secreted to the outside of the cell. So it's got to go through the endomembrane system. Those proteins that have to go through the endomembrane system, they've got a little sequence that's encoded in the gene, that's encoded in the mRNA, and that gets translated into the polypeptide. And this little sequence is called the signal peptide. It's a special sequence of amino acids that pretty much tells the cell, hey, I'm a protein that needs to go through the endomembrane system. And once that signal peptide is translated, there's a little protein that's called the signal recognition peptide. And this particular signal recognition protein, this is a particular protein that will act kind of like a little tugboat. So it recognizes the signal peptide, it attaches to it, and then it chugs along and it pulls the whole entire ribosome, polypeptide, mRNA and all, to the rough ER membrane, and then it docks. And it allows the ribosome to attach to the surface of the membrane and start to push its polypeptide that it's currently making into the inside of the ER. Once the polypeptide is finished, the signal peptide is removed, and then the protein is released into the inside of the ER so it can be polished, shipped by the Golgi out to the outside of the cell. The ribosome then dissociates, releases the mRNA, and the whole process can continue over again. So something really interesting to think about, the difference between free and bound ribosomes. What determines whether a ribosome will be free or bound is simply what mRNA it picks up. If it picks up an mRNA that codes for a protein that's supposed to function in the cytosol, that ribosome will remain free. If the ribosome picks up an mRNA that codes for a protein that's meant to be a membrane protein or a secreted protein, then that ribosome will be dragged to the rough ER membrane and become a bound ribosome. All right, a few more things. So we mentioned how transcription, the production of an mRNA, can be high throughput because you can have, multiple RNA polymerases chugging along the same gene at the same time. Well, the same thing can happen for translation. So here's an mRNA, and you can see how there are multiple ribosomes attached to that one single mRNA, and they're translating this mRNA at the same time. So 4 polypeptides are growing out of these ribosomes using just one mRNA. This whole entire structure is called a polyribosome or a polysome. Again, it makes translation high throughput. In bacterial cells, they've got one leg up on us eukaryotes. Because there's no separation between transcription and translation, they can have transcription and translation happening for a gene at the same time, multiple times. So let's take a look at this particular image. Here we've got the DNA of a particular gene, and you can see that along the DNA, you've got 1, 2, 3, 4 RNA polymerases chugging along and transcribing this gene to make their red mRNA transcripts. Remember, in a prokaryotic cell, also the mRNA, once it's made, it's done, it's ready for translation. So even before the RNA polymerases finish transcription, the ribosomes can already attach to the mRNA in creation and start translating it, which is incredible. So if you look at this one gene, at one time there could be 20 plus polypeptides that are being made at the same time for this particular gene. So definitely extremely high throughput for bacterial prokaryotic cells. For eukaryotes, we're a little bit slower just because we have that spatial separation between transcription and translation, right? So just to review the process, here we are in the nucleus, right? The DNA holds the nucleus. This is where transcription is going to take place. The RNA polymerase is going to transcribe the information for a gene to make the RNA transcript. Remember that RNA transcript in eukaryotes, it's not yet ready for translation. It has to go through processing. This takes place in the nucleus. This is where the poly A tail is added, and you can't see it here, but the 5' cap is added, and then splicing occurs, right? So we're going to take out the introns and stitch together all the exons. Once that's finished, then the mRNA is done and it can go out the nuclear pores here into the cytoplasm, find itself a lovely little ribosome, and translation can begin. For this particular figure, there's one more sort of component that's added in here that we didn't touch on too much, but over here on the side you'll notice a lot of free-floating amino acids and little tRNAs there, right? There are enzymes, for example, this amino acyl tRNA synthetase. These enzymes are responsible for taking empty tRNAs and stitching them together with the appropriate amino acids so that there are plenty full tRNAs for the ribosomes to use for translation.